century, Gregor Mendel observed a pattern in his garden that contradicted the prevailing belief in blended inheritance. He identified that traits passed from parent to offspring as discrete, stable units that remained unchanged across generations. To test this, he bred garden peas, tracking contrasting traits like pod shape and flower color. He hypothesized the law of independent assortment, the idea that the inheritance of one specific trait has absolutely no effect on the inheritance of another. When we plot a cross of two independent traits, like seeds that are both round and yellow, against those that are wrinkled and green, we expect a very specific statistical output. This 16-cell Punnett square visualizes that mathematical expectation, populating a strict phenotypic matrix of 9:3 to 3:1. This model works on paper because it treats genetic factors as independent, free-floating variables existing in a statistical vacuum. But the elegance of Mendel's math relies on a specific omission. It operates without accounting for the actual physical structures inside the cell. Decades later, evolutionary biologist Thomas Hunt Morgan needed to scale up this genetic math. To do that, he required a test subject with a much faster breeding cycle and a higher progeny count than garden peas. He turned to Drosophila melanogaster, the common fruit fly. It was the ideal organism for rapidly testing the outer limits of independent assortment. But here is what happened when early geneticists analyzed dihybrid crosses in those fruit flies. This diagram shows the expected 9:3 to 3:1 matrix actively collapsing. For specific trait pairings, the offspring heavily favored the exact combinations of their parents, while mixed traits barely appeared at all. Mendel's math breaks down because it assumes absolute independence, but physical biology dictates that many traits travel together. Resolving this statistical failure required biology to move beyond theoretical numbers and begin mapping the physical architecture of the cell. The solution arrived through the chromosomal theory of inheritance, proposed by Walter Sutton and Theodore Boveri, which finally gave genetic data a physical location. They realized Mendelian factors are locked onto long physical chromosomes. During cell division, these pairs undergo mechanical separation, moving whole gene sets. Independent assortment is mechanically true only if the observed genes are on entirely separate chromosomes. When two genes reside on the same chromosome, they are physically tethered together. We call this linkage. Anchored to the same biological hardware, they cannot separate randomly. Linked traits violate Mendel's laws because their inheritance is dictated by their shared physical vehicle, not by independent probability. The only way linked genes can generate a new non-parental combination is through recombination, a physical process where chromosome arms temporarily cross over and swap identical segments before dividing. The discovery of the chromosome took the exceptions to Mendel's rules and turned them into the foundational mechanics of modern genetics. Researchers quickly realized that recombination wasn't a random glitch. It occurred at highly predictable, quantifiable rates. This linear map plots Morgan's fruit fly data. Yellow body and white eye genes show a 1.3% recombination rate, sitting tightly together. Contrast this with white eyes and miniature wings, exhibiting a 37% rate, meaning a large physical gap. This revealed a defining physical rule. The strength of genetic linkage is strictly inversely proportional to the physical distance between the loci. Alfred Sturtevant, a student of Morgan, took these percentages and plotted them out, creating the first precise coordinate system of a genome based on the frequency of crossing over. He defined these clustered sequences as linkage groups. Since each chromosome represents a single group, the total linkage groups exactly match the haploid number. The skewed data that broke Mendel's grid actually provided the exact spatial coordinates required to chart the physical layout of the genome. With the physical constraints solved, geneticists turned back to Mendel's original logic, the assumption that every gene relationship was a binary battle between dominant and recessive. But this snapdragon demonstrates incomplete dominance, where the heterozygous state does not mask the recessive trait, but instead produces an intermediate phenotype. Crossing a homozygous red snapdragon with a homozygous white one yields a generation of pink flowers. The phenotypic ratio perfectly mirrors the genotypic ratio, one red, 2 pink, and 1 white. Furthermore, natural populations are rarely restricted to just 2 options. Multiple allelism occurs when a population possesses more than 2 specific variants of a single gene. This matrix shows the human ABO blood group, governed by 3 alleles, IA, IB, and lowercase I. In this highlighted row, inheriting IA and IB together shows codominance. Neither suppresses the other. Both express fully to produce type AB blood, contrasting the recessive double I below. Alleles are not simple biological on-off switches. They are specific biochemical products that interact dynamically to produce nuanced physical realities. There was one final assumption in early genetics that had to be dismantled: the idea that one gene controls exactly one isolated trait. We now understand the concept of pleiotropy, where a single genetic sequence controls multiple distinct, seemingly unrelated phenotypic traits across an organism. Even Mendel's pea plants contained pleiotropic effects that defied a simple one-to-one mapping. The gene dictating whether a pea seed looks round or wrinkled at the macro level simultaneously influences the microscopic size of the starch grains inside that seed. One sequence dictates 2 different physical properties. Genes function as interconnected nodes within complex developmental pathways. This systemic influence disrupts the simple input-output logic of early genetic models. Because DNA is a massive, continuous sequence of physical information, it is highly susceptible to structural copying errors. In 1901, the botanist Hugo de Vries defined these sudden, heritable changes in a DNA sequence as mutations. The most granular failure is a point mutation, occurring at the smallest physical scale, the substitution of a single nucleotide base along the chain. This animation illustrates the precise mechanism behind sickle cell anemia. A single base substitution from GAG to GUG creates a translation shift, swapping glutamic acid out for valine at the 6th position of the beta-globin chain. The physiological outcome of that single substitution is severe. Under low oxygen tension, red blood cells structurally collapse into a rigid sickle shape, restricting flow. A microscopic, single-letter typographic error redesigns the cellular architecture and triggers cascading failures across the entire physiological system. While point mutations operate on single rungs of the ladder, the system is also vulnerable to massive, macro-level structural breakdowns, categorized as chromosomal disorders. These often result from aneuploidy via non-disjunction. This mechanical failure during cell division drags entire bundles of extra genetic material into a single cell. This microscopic slide shows a complete set of human chromosomes. Notice the presence of 3 chromosomes at position 21, rather than the standard 2. This trisomy pushes the total cellular count to 47 and results in Down syndrome. Whether gaining a chromosome or losing one entirely, as seen in Turner's or Klinefelter syndromes, altering the total count of these massive physical structures warps vast networks of gene expression all at once. What began as the statistical certainty of a Punnett square quickly gave way to the physical reality of crossing over, pleiotropy, and mechanical mutation. The predictable ratios of inheritance are the visible output of an immensely complex physical system that translates raw data into biological form.